We're going to continue on with plant growth. We'll do that clicker too, so make sure you're ready to go on that. I think we left off here, looking at the idea that grasses in particular have this way of rolling up their leaves to decrease the dehydration risk when they're undergoing drought episodes. The next topic I wanted to talk about is this idea that when plants are growing, they certainly are paying attention to how much light they're getting, so the intensity, but also the quality, which is also like the wavelength or the color of light that they're getting. And so we already talked a little bit about that with the idea that the plants will bend toward the unidirectional light coming through your window, for example. So the phototropism idea, they're definitely responding to that directional aspect of the light. And so one of the questions that they had about this is, well, we know that sunlight, and if you've ever, I'm sure at some point in your academic training, you had a glass prism and the instructor put light through it and it broke into the different colors of the rainbow. And so here on the image here, you're seeing that the different colors of light are different wavelengths of light, okay? And so the longer wavelengths are in the red region, and then as you shift from the right towards the left, you can see shorter wavelengths as you get down into the blue quality of light. So I think, you know, the whole idea of the rainbow, it's red, orange, yellow, green, blue, indigo, and violet are as we move from right to left. And so it's important that you realize that it's different quality of light or colors of light depending on wavelength of light. And the way I remember that, red, orange, yellow, just the first letter, green, blue, indigo, and a small I, violet. And so the way I always remember the order of this is that red, orange, yellow spelled Roy, and that was actually my dad's first name, so that was really good. And then, sorry, I have to do it. Green Bay is victorious, although they're not victorious this year, but didn't make it into the Super Bowl. But anyways, so that's how I always remembered that, because my dad was a huge Packers fan. And so thinking about the different wavelengths and linking that to the different colors is a good idea as you're thinking about how plants are working with the light that they're getting. Do you remember what quality or color of, there's two main colors of light that the plants really like when they do their photosynthesis? Do you remember? You've had that at some point, perhaps? Anyone who's had plant fizz already? Maybe? No? Okay. Red and blue. They like red and blue. That's the quality or color that they will preferentially capture. So when I say capture, I mean that they'll absorb that quality of light. So when the leaves are busy doing photosynthesis, as the sunlight is striking the leaf tissue, they're actually absorbing the red and the blue, the chloroplast, the chlorophyll inside. They're absorbing those two colors, and they let other stuff either pass through or they reflect it. So the reason leaves look green to us is that the plants are like, well, yeah, I like red light and I like blue light, but I don't really care about green light, so I'm bouncing that back. And so then our eyes pick up green. All right. And so it's important to realize that plants pay attention to different wavelengths or colors of light, and that can do different things for them and tell them information that's really important. So this blue quality light that I have in the second bullet here, this is referring to the idea of phototropism. They were asking, is there a certain color coming from the sun, a certain wavelength coming from the sun that the plant is really cueing in on in order to do this bending pattern toward the light coming through the window. And they looked at it, and what they basically did was they were shining the light, or shining different wavelengths of light on the plant, and then they would look to see what would happen. And so what they discovered through a lot of very careful research is that there's a protein in the plant called phototropin, and it is perceiving this blue color. It picks up on that blue quality, and that's what's telling the plant to shift your auxin to the dark side of the stem, stimulate the dark side to grow so you can bend toward the light. All right. And so it was actually this phototropin protein that's kind of kicking off this whole behavior of bending and growing toward that unidirectional light. And so then they started realizing, oh, okay, there's special proteins in the plant. And since then they've discovered there's other proteins that do, they pick up on different qualities or colors of light. So it's not just phototropin, there's others out there that they've discovered. They've also looked at phototropin more carefully, and yes, it's heavily involved in kicking off this phototropism response, but they also show that it's directly involved in the process of getting the stomata to open. And so that's pretty huge for the plant. Anything that controls the stomatal movement is a big deal. And so they discovered that the phototropin is doing this as well. So what they were doing when they were, they're like, well, what quality of light? Is there a certain quality? What they did was they took the unidirectional light of different colors and shined it on the seedling and a grass seedling. And what you're seeing is that it's only in the presence of blue light that you started to see this definite phototropism response. And so that's kind of what kicked it off. They're like, okay, it's something that's absorbing the blue quality. And then they went hunting for it and found the phototropin. So since then, they've discovered there's other pigments and proteins in the plant that are cueing into different wavelengths. And then that information is important to them. They're using that as a way to respond, hopefully appropriately, to what's going on in their environment. So one huge aspect that humans have been really interested in is this idea of photoperiodism, which is basically looking at how does can the plant tell what's going on in terms of light versus dark, day versus night? And then, there's this question of, well, how come some plants flower in the spring, but some plants don't flower till fall, for example? There's this differing life cycles and differing rhythms depending on the plant species. And they started to suspect that plants were able to assess how long they were in the light versus how long they were in the dark. So a day-night thing. They were starting to piece this together. And what they discovered is that the length of the day, or if you want to think of it as length of the night, is directly related to when these plants are going to flower. And it's not all identical, as we know, because some plants flower in the spring, some in the fall, some more in the middle of the summer. But of course, flowering is pretty important to humans, because once a plant flowers, the next thing that's going to happen, hopefully, is fruit formation, which then we want to harvest and eat. And so this whole idea has been looked at very carefully and studied pretty hard. And so what they've discovered is that most plants can get grouped into different categories. So for example, there's so-called short day plants that will not flower unless the day length is shorter than some critical time period. And I'll show you some examples of what I'm thinking about with the time period. So cannabis apparently is a short day plant, the hemp plant, Cannabis sativa as well. I have in the notes section poinsettias are short day plants. Strawberries. So this is something that you can usually look up and find pretty quickly what kind of plant you might have, the species. Long day plants, on the other hand, they're just the opposite. They're not going to flower unless the period of light is longer than some critical time period. So let's look at an example here. So poinsettia is a short day plant. And so they know that in order to get those lovely red, all that red tissue, by the way, those are called bracts. They're actually modified leaves they become pigmented red. And the actual flowers on the poinsettia are actually just this cluster in this right picture here. That cluster right in the middle, those are the flowers. And then, so these are the flowers. So they're very small flowers. And so they augment those flowers with these very colorful bracts, which is just modified leaf tissue. But in order to get that look that's very popular, you have to make sure that your poinsettias get less than 8.5 hours per night of, or light, I should say, 8.5 hours of light per day. And so the one on the right here, they give it over 10 hours of light. It does not morph into the beautiful poinsettia that you'd want to go buy around the holiday season. All right, so this is a big deal for people who sell poinsettias, grow and sell those. So you better get this right, and they have to time it right so that by the time the holiday season is around, they're good to go for market. And so you can see, just like with this idea alone, how important it is to get this right. Like, you need to understand how your plant is responding to light, especially if you're trying to make a profit off of this. And so you can imagine if you own an operation like this, where you have greenhouse after greenhouse growing poinsettias for sale, you got to get it right or you're in trouble. And this was an example of a podcast I was listening to did I tell you this, of poinsettias? The guy actually went in, one of the workers, and he turned the light on in the middle of the night to try to find something in there. And the whole greenhouse of poinsettias did not flower at time. And this was a podcast I was listening to, and the interviewer said, "I bet you fired that worker." And the guy he was interviewing said, "No, I didn't fire him. He came and apologized and admitted he made the mistake. And he felt so bad, I know he'd never do it again. So I didn't fire him. I kept him." And he's been a great worker ever since. So, you learn from your mistakes for sure. That was a doozy. And so good on the employer for not firing him. I thought that was a pretty decent thing. So you have this situation where if you want your plant to flower at a very specific time, you better understand their rhythm in terms of how much light do they need for them. There's some called intermediate day plants too, that it's kind of like, it can't be too short, can't be too long. And then there's the day neutral, where that's a category where they're like, well, it just seems like this plant will flower pretty much under any day length. They don't seem to really care too much, as long as they get enough light, to survive, and they're through their puberty stage, their juvenile stage, they'll be okay. So those are some examples of these different categories. And for when they started researching all of this, they were really keying in on how do we get these plants to flower. Okay, but now we know that there's other things that the plant does besides flowering, that you need to think about this idea of how are they detecting they're in the light, how are they detecting they're in the dark. And so when this research first kicked off, it was really focused in on flowering, because we care about that, because that's how we get the fruits to form after the flowers. So they finally stumbled upon a group of pigments that are called phytochromes when they were doing all research. And it's kind of funny, one of my former advisees, Jeremiah, he got his bachelor's degree here years ago in botany. And when he came to NMU initially, at first, when he was my advisee, I said, "Well, what's your major?" It wasn't botany. And I asked him a little bit about, where he came from. And he said he grew up on a farm, and he was so sick and tired of working on the farm for his parents. And I don't want to learn about plants. I don't want to think about plants. He ended up getting his degree in botany by the end. And a lot of my research is botany focused, of course. And so he came into my research lab, and at first he's like, I don't want to do any research on plants. I'll just stick with the microbes. But eventually he got into that too. So then the reason I'm bringing him up is he published in the journal Science. He was on the team. He was the lead author on discovering the three-dimensional structure of phytochromes. So he went on to get his PhD at Madison. Did great work, all in botany. So I like to tease him about that because he swore he would not do botany. Can't help it. Plants are awesome. Turn back to them. So, all right. So phytochromes are these pigments that were discovered that can pick up on this idea of the light versus the dark. And the plant uses this set of pigments to absorb light, and then it's a really kind of a amazing system because this pigment will actually change physical shape, and that change actually is necessary to tell the plant, hey, you know, we got this thing going on, you better respond appropriately. And so they search for it in the plant. It seems like they can see it present in meristematic tissue. There's some debate. They think that there's other regions of the body besides meristematic tissues, which is like the shoot apex, the axillary buds. Okay, but so it's not. There's still a lot to learn about the phytochromes, but they have nailed this down. They know that there's two forms of this pigment. So one form they nicknamed the P sub R form. Okay, so the P stands for the phytochrome, that's the pigment. R just means that this form of phytochrome will absorb red light. So that's why they nicknamed it PR. So it absorbs red light. And when this form of the pigment absorbs red light, what happens is it will actually physically shift its shape and become what they call the PFR form. So once it shifts its shape, then they start calling it the PFR form, because if you hit that pigment with far red light, it will change its shape back into the PR form. So they refer to it as a shapeshifter pigment, which is really cool, that just changing its shape is enough to tell the plant, hey, wake up, we got some stuff to do here, we got to pay attention to this. And so it just keeps toggling, or toggling back and forth, shape shifting back and forth, depending on the quality of light that you hit it with. So this picture on the bottom is trying to demonstrate this idea, where on the left you have the P sub R. Okay, so that's the phytochrome pigment that's in the P sub R shape. You shine red light on that, and it shifts into the PFR form. And then you see this arrow off, and it says biological response. Hold that thought, I'll come back to that. If you have the PFR form in the plant, if you shine far red light on it, it will absorb that far red light. And as it's absorbing that, that stimulates it to shift its shape again back to the P sub R form. So it keeps going back and forth. It just depends on what quality of light you hit it with. You can get these phytochromes to shapeshift into these different forms. And it's reversible. They just go back and forth, on and on. So it's actually, what they're discovering is it's actually the ratio of these two. Right here. It's the ratio of these two pigments, forms of the pigment, that the plant is actually affecting. It's saying to itself, how much P sub FR do I have? How much P sub R do I have? And based on that ratio, I'm going to do this behavior. And so it might be a flower, or it might be telling it you need to grow, and I'll explain a little bit about that. So when we say it's a shape shifter, this is what we're trying to think about. So the image on the left is how phytochrome looks when it's in the piece of R form. And if you shine red light on that, it actually changes shape over to what looks like a capital Y. Okay, so it physically changes its shape once it absorbs that red light. And then we call that form the PFR form. And it's now that PFR form, it's designed in that shape. It likes to absorb far red light. So then, if you were to shine far red light on it, it would then collapse back down into that almost looks like the letter M, maybe. Okay, M to Y. If you want to think of it that way, it's fine. So it's actually changing its physical shape depending on the quality of light you hit it with. Now this PFR form, you can see there's a hand shooting an arrow off, and they're saying, you know, this is the active form. So what they mean by that is when it's in that PFR form, it is mobilized into the nucleus and interacts with proteins that interact with DNA. And so it's a way to tell the DNA to shift its gene expression and do something different, appropriate to whatever the environment is telling the plant you got to do. It's the flower, right? And so you have these protein-protein interactions. What they're trying to say there is the proteins that really tweak with, turn genes on and off. That piece of FR form is going to be interacting with that. So it's a way to send a signal to change or alter the gene expression, hopefully appropriately for the plant. So when you have these phytochromes, again, it's just this idea of which form of phytochrome are we thinking about, and understanding that one form likes to absorb red light and the other form is set up to absorb far red light. And then you can just keep playing with that, which scientists have done, to see what's going to happen when you hit the plant with either red light or far red light. Questions on the phytochrome idea? So remember, the phytochromes are pigments. The little R and the FR, that's just denoting what version of the pigment you're thinking about, the one that likes red light or the one that likes far red light. Okay, so these are pigments that we're thinking about that are capable of absorbing these different qualities. Okay, well, let's take an example here. So here's this donut is supposed to be like a time, 24-hour time period thing. So what this dotted line on the donut here is trying to show that that's the critical time period, okay? And this plant is considered a long day plant, which means it won't flower unless its light period is longer than the critical period. So that's what they're trying to show here, this tan. That means sunlight is hitting the plant past the critical mark, past the critical time period. So that's the definition of what a long day plant is. And they're trying to show, yep, it goes ahead and flowers under those conditions. However, if you create a situation where you shine light on that plant, but it's less than the critical time period, okay, so it didn't make it past that dotted line. Okay. Make it so less than the critical time period, then it will flower for a long day plant. Does that make sense? Okay. So then they started going, well, what would happen if we create a situation where we shine light on the plant, similar to what we did in the second scenario. But then we come in the middle of the night and we shine red light on the plant. What's going to happen? So they did that experiment and they were really kind of stunned to see the plant went ahead and decided to flower under those conditions. So they're like, what? But this is when they were just figuring out phytochromes and trying to piece it all together. So it's almost like when they hit it with red light, it's almost like the plant behaved as if it was getting light all the way through when the red light, you know, put on the plant. So it did go past the critical day or the critical time period, and it went ahead and flowered. So okay, so the plant got hit with red light in the middle of the night. Let's do an iClicker. Let's see if we can answer this question. And you can keep your notes open. This is open note. And you can chat. Where is my iClicker? So I'm going to start class. Okay, so let's think about this. Well, if I could get the thing to work, that would be nice. Come on. Oh, okay. I got to do this a little differently. Okay. So they came in the middle of the night. They put red light on that plant. So that remember now that plant had phytochromes in it. So what happened to the phytochrome pigment when they snuck in the middle of the night and they put red light? What was going on inside the plant? Let me see if I can do this. Can you see? Are you getting in? Okay. My computer really does not like iClicker. I'm gonna go over this DTL tomorrow. All right, so they snuck in, hit it with red light. What happened to the phytochrome? Ten more seconds. Okay. So I just clicked on results. Can you see the results now? Okay. So 58% said B, the plant ended up having more PCFR form of the pigment. That is correct. Okay. So let's go. Let me go back. Oh, come on now. Okay. So they snuck in and hit that plant with red light in the middle of the night. So what that did to the plant is it had some piece of our form and tissue that part on it. You start hitting that with red light, it absorbs the red light, starts to shift into that Y-shaped piece of FR. So if you're a long day plant, that's actually a signal. Yeah, go ahead and flower. Go ahead and flower. When you come in the middle of the night, it's like it's like, oh, I've been exposed to light. And it's all this swath of time. And so it starts to go ahead and flower. So when they discovered this, they kept playing with it. Like, then they came in and they're like, well, let's hit it with fire red light next. Now what's gonna happen? So what do you think would happen? They came in and like, let's hit it with red light, and then a half hour later, let's come in and hit it with fire red light. So if you hit it with fire red light, what's happening to the cytochrome? Which versions that start increasing? PR. Yeah. So the PR form goes up, and that's telling the plant no. Okay. And so you can do that over and over, which they've done. And you can like only control whether or not that plant is going to flower just by shining a little bit of red light or far red in the right pattern. So basically, and this is another thing that they'll say about it, they'll say the plant only remembers the last quality of light it got hit with. Okay. And so here it got hit with red light. So it came in, let's say it was red, then far red, then red. And it's like it's thinking to itself, oh, I've been exposed to light this whole time because the red light converted my PR to PFR and telling me to fly. And so you can do this over and over and get these amazing results. I think they gave them maybe it was minutes of time, and they've even done some research looking at even like moonlight, which is like reflected sunlight. Like even that on a bright full moon night, it can cause shifting in the phytochromes, which is interesting. So it's not necessarily the super bright light that you have to have shining on the plant to affect it. That also makes you think about like landscaping lights or lights along the street. That's another thing too. It's like, well, what's happening to the phytochromes under, with the plants exposed to that? And it does affect them. If it's the right quality of light, it does affect them. So landscape architects need to know that whole thing. And so you can do these experiments and they're pretty, like, you can get this to happen. It's pretty consistent. And so, okay, so with red and far red, let me just, let's think about this idea. So let's think about a forest ecosystem. And when you think about sunlight hitting a forest ecosystem, so you think about a more mature forest ecosystem that has the upper story plants, the really tall ones, and then you have mid-story, and then you have understory. And so the ones that are lower in the canopy layers, they're shaded, right? I mean, we know this. We've not been out hiking in the woods. Well, one thing you need to appreciate, going back to when I was asking you what quality of light do plants want for photosynthesis? What were those two colors? Red and blue. Okay. So let's think about it. We have maybe it's a gigantic oak tree that is in the upper canopy and it gets hit with light, full on light in its crown. So it's going to take the red light and it's going to absorb it in its chlorophyll, which means the lower plants aren't getting as much red light. Think about the oak tree hogging all the red light. I like the red light. I'm taking it. And they are taking as much as they need. And some might filter down, but a lot of it's getting absorbed. So what that means is the mid and understory plants, they're getting less red light and they're actually getting more far red. Because the plant's like, yeah, far red, not so much. I'll let that filter through. I won't absorb so much of that. It's really the red I want. So what happens if an understory plant is like, okay, I'm not seeing much red, I'm getting a lot of far red. What's happening to its phytochrome? What starts to build up? Which version of phytochrome? PR starts to build up in its body. So it's like, oh, getting a lot of PR, getting a lot of PR. It must be getting shaded out. And so when a plant has a lot of PR formed, it's telling the plant, you're getting shaded out. Now, if you're an understory plant and you're adapted to shade, that might be totally fine. The plant's saying, I'm getting shaded out, but hey, that's okay. I'm an understory plant. That's what I've adapted to do. Everything's cool. But if you are a young seedling, like maybe you're an oak that needs high light, and your body's telling you you're in the shade, but I'm a highlight plant. Uh-oh, what might you want to do in response? Grow, right? You might go, hey, where's the light? I gotta get to the light. I'm a light highlight plant. I want light. And so depending on the plant and its lighting needs, it may be totally fine with being shaded. On the other hand, it may be like, ooh, I got a direct energy to growth. I need more sunlight. And so these ratios of PR to PFR, very, very important for the plant to figure out, am I light or am I shade? And then they might need to respond by growing if they're shaded out. And, you know, it's like, well, I need to grow in order to get access to light. So plants can do that. They also have found just the overall plant development. So thinking about the form of the plant body. So for example, if its nose is getting shaded out, it could grow when it needs light. It could grow taller, or maybe it's going to grow more lateral. Like maybe if it just reaches its branches out a little bit this way, it'll catch some sunlight. And so you can see plant development shape can vary depending on what the phytochrome ratio is in the plant body. They're seeing that the phytochromes seem to play a role in the plastids and getting the plastids to kind of like, okay, you're destined to be a chloroplast, you're destined to be a chromoplast. Seems like phytochromes are involved in that process. They're still working out the details. And then just certain pigments seem to be dependent on information that the phytochromes are providing. Like, should we go ahead and kick into a lot of anthocyanin, which is a pigment? Anthocyanins are pigments. So anthocyanins are in beautiful, some of them kind of bluish or purplish flower colors or fruit colors. And so again, that's, and then the detection of shape, as I mentioned. So these phytochromes, the ratio of how much PSABR do I have, how much PFR do I have, and the fact that it can actually change shape. It's been pretty phenomenal work that's been going on, and a lot of surprises that something like that just changing shape, and that can really drastically alter how the plant will respond to whatever signal it's getting. So phytochromes are what we call photoreceptors. That's a fancy word. And we now know there's a bunch of photoreceptors in the plant. So phytochromes, that phototropin I mentioned, that's a photoreceptor. So they gave it that name because it's a protein, and then there's usually some special other molecule attached to it called the chromophore. And so, but all these photoreceptors, what they do is they absorb a certain quality of light, and then that will allow them to change their shape. And it then allows for some kind of signal cascade ultimately to the DNA, telling the DNA, the gene expression to, change. And so they call them photoresponses in the sense that it's all driven by the quality of light that these different photoreceptors are getting hit with, absorbing, and then that changes their shape. And then it could, if it's the right shape, that could then lead to a change in gene expression. And so it's really, they're using light as a signal. And then that kicks off the signal cascade all the way to the DNA. telling the DNA to shift its gene expression. And so phytochromes were the first, I think, to be discovered that they shape shift like this. And since then, there's been these other scientists that have come along and they are like, oh, there's other photoreceptors out there too, and they also can alter their shape, and that can give important information to the plant. It changes its gene expression and allows the plant to adapt to its current environment. So again, this all dependent on what quality or color of light. In this case, for phytochrome, it's red or far red light. Questions on phytochrome? I know I just threw a lot at you. And the first time I learned about phytochrome, I was kind of walking out of there scratching my head. So please spend some time. If you have time right after class, you could sit down with this and just look it over, even if you just have 5 or 10 minutes. I encourage you to look this over and start getting this, getting your head wrapped around it. Good question. So red and far red light, when we look at it, let's go back to this one. Wow, this one. Go to this one. So red and far red is more like 800 nanometers wavelength of light, and red is closer to the sun. And so it's just a different wavelength coming from the sun of light energy. And to our eye, we can break that out into different colors of light when our retina perceives them. I thought I saw another hand go up. Yes. But yeah, it has to do with, and we'll get into that topic pretty quickly. It has to do with how the chlorophyll molecule is designed to absorb certain qualities of light for photosynthesis. And of course, photosynthesis is hugely important for the plant. And so when they're getting hit with sunlight, so much of that sun, the red quality goes for photosynthesis. And then the far red is somewhat involved in photosynthesis, but not as important. And so that's what tends to get trickled through to the understory. Other questions on phytochromes? Yes. So phytochromes, when we talk about those, it's red and far red, but there are other photoreceptors like the phototropin response to blue light. There's even photoreceptors that respond to ultraviolet light, which is more like 400 and what else? Cryptochromes, those are photoreceptors in plants, and they all have their like specific wavelength that they're adapted to absorbing. Like that's their favorite wavelength. So when you think about plants, and we've been talking a lot about plant growth, how they respond to their environment, their use of their hormones to coordinate everything and hopefully respond and behave appropriately to their environment. When you think about it though, with their plant growth, and you think about humans, let's say, compared to plants, like we have all these different organs, right? We have a brain, we have lungs, the liver. So, if something happens where our lungs get taken out of our body, we can't make it, right? I mean, we're done. But a bunny rabbit or a pack of bunny rabbits, what's a pack of bunny rabbits called? I don't know, but they could chomp their way on that plant and practically annihilate the plant, and they can survive. So they're so different from us in that regard, right? I mean, if something comes along and chomps our brain, we're done. So plants have, primarily because they are so stationary for their life, right? They can't pull out a gun and fight off. They can't run away. And so they decentralize so much of the stuff that we don't decentralize. We put everything in organs and have very specific regions of our body. They have decentralized. And that allows them to get chewed down to virtually nothing. And they're like, yeah, I'm okay. I'll just grow again. is really quite amazing and such a different lifestyle compared to, you know, humans in that regard. And so this idea of decentralization, they breathe with their whole body, see with their whole body, feel with their whole body, and evaluate with their whole body, spreading each function over the entire organism as much as possible is the only way to survive predation and damage. And plants can do it so well that they can even withstand removal of much of their body without losing functionality. I mean, that's pretty wild to think about. And so their ability to, I mean, I just love the way, you know, they breathe with their whole body. And we get into, when I teach my advanced botany class, we get into this quite a bit on how they're doing this. But part of the reason they're so good at this and able to, you know, decentralize, and then if somebody comes along and decimates, you know, eats up most of the tissue, they also have this capability called plant totipotency, or they're totipotent organisms. And basically what that means is just, you know, a little couple of cells of a plant cell is enough for it to regrow and differentiate into a plant all over again. And so we take advantage of this in tissue culture technique. But all you need is a few living cells, and you can coax them through the pathway. Just take a few cells from that, and under the right hormone concentrations and the right nutrients in a tissue culture process, you can get those few cells to grow, 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 grow. Then you hit them with a different ratio of hormones, and then they go, oh, I'll start forming roots, I'll start forming shoots, I'll start forming leaves, and you're back to your plant. And so whether it's a bunny rabbit or a herd of bunny rabbits doing this, or in the lab, this ability of them to have these cells kind of figure it all out, like, okay, I need to grow, and then I need to differentiate into different things that I wasn't doing before, you know, because this is a template. We took template cells. And those template cells are like, okay, some of me has to stop being a template, but some of me now has to do shoot, leaf growth, some has to do root growth. And that's a phenomenal amount of gene expression shifting, and it's amazing. So give them a lot of credit, and think about that when you're looking at some of these nature shows, you know, and all these animals chowing down on plants, and yet they'll just regrow. So decentralization. Okay, we'll stop there and continue. Well, we're going to get into a new topic on plant propagation. In the lab this week, you're going to go to the greenhouse and propagate plants. So on Wednesday, I'll start that topic in lecture. Yes.